The spermatozoon. The spermatozoa or male germ cells are developed in the test and are present in enormous numbers in seminal fluid. Each consists of a small but greatly modified cell. The human spermatozoon possesses a head, a neck, a connecting piece or body, and a tail. The head is oval or elliptical but flattened, so that when viewed in profile it is pear-shaped. Its anterior two-thirds are covered by a layer of modified protoplasm, which is named the head cap. This, in some animals, for example the salamander, is prolonged into a barbed, spear-like process or perforator, which probably facilitates the entrance of the spermatozoon into the ovum. The posterior part of the head exhibits an affinity for certain reagents and presents a transversely striated appearance, being crossed by 3 or 4 dark bands. In some animals, a central rod-like filament extends forward for about two-thirds of the length of the head, while in others a round body is seen near its center. The head contains a mass of chromatin and is generally regarded as the nucleus of the cell, surrounded by a thin envelope. The neck is constricted in the human spermatozoon than in those of some of the lower animals. The anterior centriole, represented by 2 or 3 rounded particles, is situated at the junction of the head and neck, and behind it is a band of homogeneous substance. The connecting piece or body is rod-like and is limited behind by a terminal disc. The posterior centriole is placed at the junction of the body and neck, and like the anterior consists of 2 or 3 rounded particles. From this centriole, an axial filament surrounded by a sheath runs backward through the body and tail. In the body, the sheath of the axial filament is encircled by a spiral thread, around which is an envelope containing mitochondria granules, and termed the mitochondria sheath. The tail is of great length and consists of the axial thread or filament surrounded by its sheath, which may contain a spiral thread or may present a striated appearance. The terminal portion or end piece of the tail consists of the axial filament only. Krauss gives the length of the human spermatozoon as between 52 and 62, the head measuring 4 to 5, the connecting piece 6, and the tail from 41 to 52. By virtue of their tails, which acts as propellers, the spermatozoa are capable of free movement, and if placed in favorable surroundings, for example in the female passages, will retain their vitality and power of fertilizing for several days. In certain animals, for example bats, it has been proved that spermatozoa retained in the female passages for several months are capable of fertilizing. The spermatozoa are developed from the primitive germ cells, which have become embedded in the testis, and the stages of the development are very similar to those of the maturation of the ovum. The primary germ cells undergo division and produce a number of cells termed spermatogonia, and from these primary spermatocytes are derived. Each primary spermatocyte divides into 2 secondary spermatocytes, and each secondary spermatocyte into 2 spermatids or young spermatozoa. From this, it will be seen that a primary spermatocyte gives rise to a 4 spermatozoa. On comparing this process with that of the maturation of the ovum, it will be observed that the primary spermatocyte gives rise to the 2 cells, the secondary spermatocyte and the primary oocyte to 2 cells, the secondary oocyte and the first polar body. Again, the 2 secondary spermatocytes by their subdivision give origin to 4 spermatozoa, and the secondary oocyte and the first polar body to 4 cells, the mature ovum and 3 polar bodies. In the development of the spermatozoa, as in the maturation of the ovum, there is a reduction of the nuclear chromosomes to 1/2 of those present in the primary spermatocyte. But here the similarity ends, for it must be noted that the 4 spermatozoa are of equal size, and each is capable of fertilizing a mature ovum, whereas the 3 polar bodies are not only very much smaller than the mature ovum, but are incapable of further development, and may be regarded as abortive ova.